Okay, well, thank you everyone for turning up. This is what I'm going to talk about today. But before I get into that, I'd like to say a few words about SRUC, where I'm employed, because I suspect that will be a new organisation to many people, perhaps. Scotland's Rural College. It's large, it has a turnover of about 80 million, and there are three strands to that. There's a research strand, a consultancy strand, and an education strand. I'm in the research strand. That has an income of about 21 million, a third of which comes from the Scottish Government. And drilling down a bit further, you end up where I sit in the Directors Research Group. I'll go through this quickly because it doubles as a slide of acknowledgements. So here's Wayne Powell, the director, Dave Marshall, formerly of the James Hutton Institute, who many of you may know, myself, Ian Dawson, now working for the CGIAR Agroforestry Institute. The four of us are kind of part-timers in terms of our research contribution. So I like to think if you put us together, we kind of make one old man, which is more than enough. Over here representing youth, we have Rajiv Sharma, who's talked to this group before, CJ Yang, who's also talked to this group before, and Stephanie Bajay, who is, we'd love to have with us, but she's in Brazil at the moment. And the focus of the group is crop genetics, plant breeding, and quantitative genetics. So to sell seed, a variety has to be added to the national list. And for that purpose, they have to be classified as distinct, uniform, and stable. So different from other varieties, consistent within the variety, and stable across seasons and sites, seasons and sites. And that's assessed mainly through a modest set of morphological traits over 2 years. And they must also be classified as suitable for cultivation and use, so-called VCU, and that's assessed typically in trials over 2 years. And the intention of this system, which has largely been achieved, is to give breeders a return on their investment through a royalty payment on seed, which seems reasonable, to protect growers from poor product through a seed certification scheme and knowledge of the results of those independent yield trials, which also seems reasonable, and to avoid stifling innovation through something called breeder's privilege, which entitles one breeder to use another breeder's varieties in creating new product. If this was a patent system, that wouldn't be possible. And here's some breeder's privilege in action in German winter wheat in this recent paper. The 133 varieties that this group looked at have been classed into those where the breeder produced the variety but used as parents 2 lines from other breeders, and that's the number at the top, 81. The breeder produced the variety but used 1 line from another breeder, of which there were 40, and there's only 12 varieties where the breeder owned both the parents and the progeny line. So the vast majority of lines released in the period these guys looked at in Germany involved the use of parents outside the breeder's own germplasm, if you like. So in trying to describe what I think is going wrong with the US, I'm going to plagiarize heavily the work of CJ, which is has been accepted. Its release in Communications Biology is imminent, and that also allows me to acknowledge the contribution of Joanne Russell, Luke Ramsey, and Bill Thomas from the James Hutton Institute into this work. The problem, though, before we go into detail with some of CJ's results, is it's basically near impossible to get the system to change. And in particular, the use of molecular markers, which you kind of think was a no-brainer, has been severely restricted. But in some cases, at least, and in particular in spring barley, as we'll see, The current system is now at the stage where it looks like it's hindering rather than helping the breeder in some cases. So you are allowed to use markers, but in a restricted sense. And the one of the uses is you can use them in a sort of standard way to look at differences between markers and classify those differences. But that has to agree precisely with the same differences that you would get if you use the DUS traits for that purpose. So this is some barley data. There's, I think, 710 varieties there. There are 28 DUS traits for barley, and you can see that the correlation is pretty poor. Even with large numbers of markers, you don't rise above that threshold of about 0.6. But what we've done is look at the DUS traits themselves and split them into those with high heritability and those with low heritability. And you can see the blue dots there, the higher heritability traits, give you a little kick up in correlation, but the number of these 28 DUS traits of low heritability and are really not doing a job in terms of assessing differences between varieties compared to the markers, though the traits have precedence over the markers, I should say, again. We and others before us have also looked at reducing the number of markers. Do you really need 1,000? Can you get away with a smaller subset? Yes, you can find subsets that discriminate amongst all the currently available varieties, but if you breed in silico from those varieties and go ahead a generation, you generally find they don't do so well after all. So for this purpose, we would recommend using 1,000 markers, and current costs for doing that are really quite low anyway. The second use to which you are allowed within the system, the current rules, to put markers is if you can find one or more that act as perfect substitutes, surrogates for the traits themselves. And the obvious way to do that, and this isn't original, is to carry out a GWAS of the trait and see what you find. So these are CJ's results, a subset of the 28 varieties. And you can see, for example, for trait number 19, there's no problem here. We've got a clear marker here. We might have to use that one as well, but we're going to get very good discrimination with these two markers, or very good agreement with whatever trait number 19 is. Same for trait 22, perhaps not so good, but not that bad for trait 23, but nothing at all for trait 18. If we look at trait 18, ignore the fact it's now labeled as trait 17, we can see why. The estimate of heritability for this trait is 0.26, which isn't great. And you can also more or less see why the heritability is so low. The traits are scored into those categories given in the picture there. And, you know, I might be able to discriminate a score of 1 from a score of 7 or 9, but the chance of discriminating a 3 from a 7 is really not that great at all. And in fact, the heritability of many of these DUS traits is low. So the average is 0.5, which isn't great for a trait that is meant to behave to get these varieties listed and allow someone in Italy to get the same results when they look at a variety as someone looking in Scotland. The minimum is 0, which is really worrying, and the maximum is 1, which is if these will be the traits that are effectively Mendelian. Things are worse because if you look within spring barley or within winter barley, the averages drop a bit more, and generally that's what we're interested in doing, discriminating between spring, within spring, or within winter. We know what a spring and a winter barley are. Another way of looking at this is to look at the repeatability of traits between expert scorers. So the initial scoring of these traits is done by NIAB, but the Science Advisory Service for Scottish Agriculture also carry this out on the lines that pass the US through the NIAB system for their own purposes. And I'm not at all criticizing either of these organizations. They have experts, highly skilled in measuring these traits, but they don't agree perfectly at all. I mean, they get perfect agreement on average about 65% of the time, but they get quite big scored differences, worryingly big, on quite a large number of occasions. And that's not that surprising, I think, given those low heritabilities. And we're running out of options. So new varieties have to slot in as looking different from the existing varieties. There are only 28 traits. Some of those traits are not good. If you look at the consequence in terms of the average distance between varieties over time, it's declining. So here we've got a rolling mean of average distances between 20 spring barleys, and going along with, we'll drop the oldest variety, bring in the most recent variety, recalculate those distances, and away we go between 1982 and 2015. And you can see that by the time we're down into these more recent years here, the last 10 or 15 years perhaps, things are sort of bottoming out at very low distances on average between the varieties. And these are the winners. These are the varieties that got through the DUS system. There will be varieties that failed to get through. So this is a sort of optimistic picture that we're looking at here, rather than a wholly unbiased picture. And finally, these markers, or some of them, are correlated with yield. And the sort of pure DUS system for cereals anyway treats these markers as though they're independent of yield, they're independent of the breeding process itself. If they are correlated with yield, then they're getting in the way, basically. So for the 28 traits here, we have the phenotypic correlation with the data that we've got, given in the bluey green color, and that's quite small. But if you look at the genetic correlation, some of these are really quite large, and so I'll focus obviously on this one here, where the estimated correlation with a large standard error is 1. That particular trait is this one, ear shape. It's classified into those 3 categories, again with very low heritability. So really, I think we're at a position, particularly in spring barley, but you know, why not for other crops as well? Why aren't we using markers? This is crazy. Some of us have been arguing this for decades, and it's not that difficult to impose a marker system. You basically, depending on what you're looking at, you have a threshold. And so, for example, for distinctness, you look at differences between a new variety and existing varieties. If the difference falls to the right of the threshold, you would accept it as being distinct from the existing varieties. Uniformity, you just look at differences between individuals within a variety. You don't require or expect all individuals to be identical. That's not consistent with the way varieties are produced, even for inbreeding crops. So it's just a question of where you draw the threshold again. And heterogeneity is something that you would assess in terms of differences between different bulks of seed or across years in a similar sort of manner. So that's all I'm going to say about DUS. I'd like to say something about the VCU system, the system of testing varieties, which gets them onto the national list, but incorporate into that the additional years of testing that are required really before any variety will start selling seed. That process of moving from the national list to getting recommended list status, which will take up to an additional 3 years. And the description of the national list, recommended list process was well set out by Desmond Patterson and Valerie Sylvie in 1980, which was the year I started work. And it's a great paper. It influenced me, and it was world-class at the time. And the UK had a world-class system for carrying out this process. And it's still pretty good, but it hasn't really changed much at all since then. It may not have changed in other countries that much either, but most notably in Australia, it's changed a lot. Within this paper, the Patterson and Sylvie paper, the quote at the bottom is that much remains to be done in designing series of trials and interpreting the results. So the thing is, much has been done. It just hasn't got through into the system, and it really should be in the system, I feel. So I'm going to try and illustrate that in the next few slides. So this is a section of the UK recommended list for the forthcoming season, starting 2021. And it's nicely laid out. There's a list of varieties here. There are more varieties than shown there. And down at the bottom here, we have their yields estimated for a UK average, and then for 3 regions within the UK, East, West, and North. And I'd like to draw your attention to these 2 varieties on the right-hand side here, which have been classified as new, drawing the attention of the users of the list to this status. They've just come in. There they sit. But for the northern region, a recommendation is surrounded by square brackets. And this is indicating that for the northern region, there's less data. So to treat the results for that region with caution. These are the regions, and I guess the UK is a pretty small country, but these are still fairly variable geographic and climatic regions potentially. We're arguing that a recommendation for the eastern region, for example, would be saying that a variety grown up here, say in Teesside, is going to perform relatively the same compared to its partner varieties as something grown down here in Kent. I'm interested in what's going on in the north because of that, those square brackets, if you like. And what I've done here is take that same recommended list, taken all 33 varieties, and just correlated the yield estimate for the north with that for the east and the west. And it's 0.7 and 0.6. So not great, but okay, not so bad. If I only take the varieties that have been tested in 5 years, then I'm down to 25 varieties, and the correlation jumps up a fair bit, really, from 0.7 to 0.8 and from 0.6 to 0.7. The reason for that jump is because those varieties with limited data, only over 3 to 4 years, there's only 8 of them, and the correlation of the north with the east and the west is really quite poor there. So it's right to say that the varieties with less than 5 years worth of data are not well assessed in the north. But there's things we can do about that. And we could have done this in the 1990s for sure, but we're still sort of lagging behind. I don't have the data to illustrate that with the for the national list, but I've taken a GWAS panel to sort of show you the same thing. This had 386 varieties, much more variable than you would see in the recommended list, but only 6 sites, many fewer. So there were 15 inter-site correlations there. And this thing, the blue, the best linear unbiased estimate, is the way that the recommended list is run, basically. And it gives an average correlation of 0.48 and quite a low minimum, 0.23. We just switch, and it's not a big switch, from using blues to BLUPs, best linear unbiased predictions. The average correlation between sites is now 0.8, and the minimums come up a lot, it's 0.71. And what BLUPs do is borrow information from the other locations. So if you had 2 locations where varieties were performing very similarly, in an extreme case, you might say, right, I'm just going to combine the two and take the average. Equally, if you had two locations where the varieties did not correlate at all, you'd leave them alone. You wouldn't try and average them in any sort of way at all. BLUPs take a sort of weighted view across all sources of information, all the site information, to come up with the figures there. And what that potentially allows you to do is give more accurate regional recommendations. So perhaps we could give the Scottish growers better recommendations than they're getting at the moment. And you could potentially split up those regions into smaller blocks, potentially going down to a single site if that looked like it was the worthwhile doing. But there is something worth doing there that we're not doing. And just as you can use BLUPs to borrow information from other sites, you can use BLUPs to borrow information from other varieties. So related varieties perform similarly. You can estimate that relationship cheaply with markers, with the same set that we're sort of advocating you could use for DUS purposes, and produce a prediction of the yield of varieties that you've never seen before. And that seems to work. So this is the barley data, again, spring barley data, and it's some finished work with Keith Gardner from NIAB, and we've taken 5 years worth of data, akin to the recommended list, and predicted the performance of the new varieties in the 6th year. So that's like predicting the 1st year of national list trials. The correlation is 0.4, which is not great, but it's pretty respectable. It looks much more respectable when you think that the correlation between successive years is only, phenotypically, is only 0.457. So there's a lot of information out there that could be used from related varieties to either improve precision, or you could use that to consider dropping a year's worth of testing to get onto the recommended list, or you could consider using it to maybe even drop a location or something like that. But the information is there, and at the moment it's being wasted. Except some breeders are doing this, and I know that because I've seen it, and I saw it about 10 years ago. So it might even be all breeders now. And the outcome is that some of the breeders have a more accurate version of the recommended list than the recommended list itself. So, you know, kind of think it's time that we should be doing something about that. There's other improvements that could be made to the VCU recommended list system. I'm not going to go through those except mentioning my favorites at the end, which is the analysis is just plain wrong for untreated trials. You get biased estimates of variety effects out of it. And again, we've known that for years, but the system doesn't change. So in summary, you know, for DUS, we've got to start using markers sensibly. The current system is stifling innovation, I feel, in some aspects rather than encouraging it. And for VCU trials, we really ought to start adopting some of these more modern methods. They're not that modern, really, many of them, but you know, let's try and catch up with the Australians a bit. And that's it. My rant is over. Thank you very much for listening.
Thanks. Thanks very much, Ian. That's very interesting stuff. So, if anyone has any questions for Ian, please put them into the question box, and I will relay them to him. So, I mean, I must admit, Ian, this isn't my area of expertise, but let me ask you first of all. I mean, you've got a lot of suggestions how this process could be improved, and how are there open discussions about this process, or is it a bit of a closed book at the moment? What's the option for these changes being adopted?
I think I mean, there's sort of internal discussions in various organizations, including SRUC, but and there were, you know, when I worked before at NIAB, and there were things were discussed there as well, but there is a lot of resistance to change, I think. So people fall into two camps, of which, you know, I'm obviously in the, can we reform these systems that have been in place for decades, and the camp that think, no, leave things alone. But you know, I think for some crops, such as spring barley now, we're at a position where something really has to start changing, and the existing trait set shouldn't be given primacy anymore. There's no harm in using them. If you've got good traits and you can score them and they have the uses, that's fine. But to give traits with, you know, a heritability of 10% primacy over 1,000 markers is just nuts.
Mm-hmm. Okay, so we have a few questions coming in. So first of all, Catherine Howarth asks if there are enough trials to make more regional recommendations.
I don't know is the truthful answer, but I suspect certainly for winter wheat there probably are. It's easy enough to test this, so there's a lot of historical data out there. Once you've got access to the historical data, you can do the same sort of thing that I was describing with Keith. You know, you can make you can run through the data, reanalyze it, make a sort of regional recommendation on the basis of that, and test it against the what actually happened in subsequent years. I suspect for oats and grass that may struggle a bit more, which I know is Catherine's interest.
Okay, so a question from Tina Barsby, and she asks, how would you propose that you get the system changed, since there is huge resistance within the system?
Well, I think well, there's two strands to that. One is to get access to the data and funding to be able to do a good job of reanalyzing it and demonstrating that there's improvements that can be made. And the second is to lobby the directors of major institutes, I guess, Tina. But I know again, when we were at NIAB, we tried to propose that the system for designing trials changed a little, and we could demonstrate that there were improvements there, but that never got taken up either.
Mm-hmm. Okay, so another question from Catherine, and she asks if you could explain a little further about the issues with the untreated trials that you mentioned.
It's because if you have a 5-year period or any period and the resistance breaks down of a variety breaks down in somewhere within that period, the consequence of not acknowledging that resistance breaks down is that it sort of bounces the variety performance up for new varieties coming in and being tested against it. And to compensate for that, it pushes the year effect down. So the outcome is, if you look over a long enough period of years, it looks like the environment's declining drastically, and the breeders are doing a stunningly good job of producing new varieties which compensate for that. And if you compare rates of increase of variety performance in untreated trials with that which you would see in treated trials, it looks like things are going much better in the untreated trial series, and that isn't the case. I mean, you can sort of there's a paper of 2011, 2012, something like that, which reanalyzed the national list recommended list system for a range of crops, and that illustrates what's happening. You can see it. It's really a very marked effect, and there's also a sort of cartoon figure to explain what's going on in that paper.
Okay. So a question from Phil Howell, and he says, your examples are cereals with relatively large trial matrices. Do you think it'd be worth trying this in crops with fewer locations to increase robustness?
I guess for the locations, yes. I mean, you've got nothing to lose, basically. I mean, with the GWAS panel, there was only 6 locations, and that worked well, though how much of that depends on there being a large number of varieties there, I'm not sure. The prediction stuff would start to fail then. You need a decent number of varieties to genotype to be able to make those predictions and gain information from relatives. Site numbers, I don't know. Again, perhaps for something like wheat, that's something you could look at, because you could start dropping sites out of the matrix and just work with the others and see how far you could get, so that those results might then translate to less tested species like oats, where there are fewer crops that are available, fewer trial sites are available.
Okay, so a question from a little bit linked to that from Peter Wally. So if he says, if the markers are used, would this not require an agreed panel to be used on all these new varieties and also a selection of the older varieties, and who therefore would pay for that genotyping?
Well, I think taking the payment bit first, it would substitute for, in the extreme, it would substitute for the existing DUS system, and it would be cheaper, so it would save money. And the agreed panel, possibly, yeah. I'm not sure to what extent you'd have to have, it would have to be a sort of unbiased genome-wide set, I think. I wouldn't think there was huge difficulties in agreeing that. What I'd be wary of, ending up doing the same sort of thing, ending up in the same position with markers as we're in with these traits, that especially if you decided to try and use a very small set of markers. So you said, right, we're going to agree to use these 100 markers, and it became apparent a few generations of breeding further on that they had become inadequate. I think, the ideal would be everything just gets sequenced, but we're way off that yet. I can't imagine that being a huge sticking point, other than the cost of genotyping when that to make a change to that. But the cost of genotyping are becoming substantially cheaper than the cost of phenotyping.
Mm-hmm. So John Bannock kind of has a question related to that a little bit, and just I guess a more global question: what do you think is hindering the adoption of markers in this system?
I don't know. I mean, it seems, I don't know. There's a lot of resistance within, maybe it's people whose jobs and lives possibly have been devoted to these traits and working with them and resolving problems in their use and all these sorts of things. So that if someone, not me, what I'm saying is not original at all, breezes in and says, let's just get rid of it and use markers, it's much easier, if probably less skillful in a sense. It's almost like, people were very skilled with using slide rules, and then calculators came in and just wiped them out. And those of us who'd got some use, or could more or less manage a slide rule, were sort of almost a bit peeved, because there was a bit of an art in trying to get them to work properly. But that's progress.
Yeah.